It is antimalarial use of Malagasy plants is clearly correlated with performance of antimicrobial bioassays. To just say a word or two of background, there are two really different approaches to both using plants as medicine and researching the health benefits of plants. You can use plants as traditional medicines with multi-component extracts, and you can study the efficacy of those extracts. Or you can use plants as a source of lead compounds for drug discovery. And there's certainly overlap between these two categories, in as much as most of the early pharmaceutical drugs were derived from plants usable for the same purpose. However, just as there are a lot of medicinal plants that can't be reduced to a single compound and put in a pill, there are quite a few potential drugs in plants that aren't found in plants with the same utility. The big example is vincristine, which treats childhood leukemia, derived from a plant, or as a periwinkle, that can't be used to treat cancer. These are two totally different approaches to research, and natural products discovery, going out and collecting all of these plants out there to see whether they might have any lead compounds suitable to make new pharmaceutical drugs, is a big area of research, although one that waxes and wanes. I was involved for several years with the International Cooperative Biodiversity Group project in Madagascar. The ICBGs were US-funded programs that combined government, academic, and corporate research partners to look for useful compounds and had a conservation purpose. So the three major components of the project it included collection of materials, screening, and conservation. David Kingston at Virginia Polytechnic and Dr. Etienne Rakutube, before his retirement was forced by illness, were the leaders of this project. And several thousand plant specimens, soil samples were collected at 5 conservation sites. Community-based development programs and conservation programs were set up, and numerous new compounds were published in the chemical literature as a result of the lab work. So in these regards, it was a very successful program, even though to date, no pharma drug has emerged from it. And that is a very typical thing in natural products discovery programs. Some people have the impression of bioprospecting that anytime someone picks a dandelion out of your yard, he's going to be making $1 billion drug out of it. The usual outcome of discovery programs is that they fold up without ever having gotten a product to market. Even the initial hit rate, the percentage of plants that have something interesting enough to pursue under the current single molecule paradigms is low enough that very frequently the industries that might be interested in funding this research say, well, this is a waste of money, we're gambling a lot, not likely to get anything. So how can we make it more efficient to get people out there looking for the useful things that we know are out there in nature? Well, one of the suggestions is, if we didn't just screen at random, but we screened according to ethnobotany, why traditional healers have been spending the last thousand years or so looking through the flora for things that are bioactive. Therefore, surely medicinal plants should be more likely to hit in assays looking for bioactivity. And indeed, I list up on the slide 3 examples of several studies that tested this directly and found that ethnobotanically selected plants were indeed more likely to perform well in various bioassays than plants selected at random. Now, the subject of malaria in particular is of interest. There was a publication a few years ago by a guy named Stephen Morantz. He observed that despite the plethora of medicinal plants used for malaria in Africa, did the sound just cut out? Okay, you guys can hear me? That's going to look good on the film, sorry, Cassie. Despite this great number of anti-malaria plants, no drug comparable to artemisinin had been developed for many of those plants. And his argument was, well, that's because those plants aren't really going to work. They aren't very potent. And his argument was that when you have really high prevalence of malaria, as you do in Africa and portions of Africa as compared to Asia where Artemisia annua was used, resistance will develop rapidly to any drug that quickly kills the parasites. Now, we tend to think that the reason plants have multiple active compounds like quinine and cinchona isn't the only active alkaloid, it's actually a suite of several. And the very reason those things exist is to slow the development of resistance. But he thought when there's constant re-exposure in a large population, no matter how many active compounds there were, the parasite would evolve resistance quickly. And he used the fact that childhood mortality rates dropped after farmer drugs for malaria were introduced to say that the original drugs weren't effective at killing parasites. Instead, he argued what the traditional medicines would do is improve host resistance, improve host ability to fight off the disease, which is something that the parasite can't really evolve resistance against. As supporting evidence, he offered 3 studies screening African antimalarial plants and bioassays in which they had low hit rates or in which few active single molecules were found, which is 2 entirely different things. Another thing that he didn't comment on but could have commented on is that there's a philosophical argument that you don't actually want to kill parasites if you're living in an area where you're going to be reinfected again in a few months anyway. Total parasite kill is the only thing that a Western physician treating an unhappy tourist would tolerate. But if you're going to get sick over and over again, you'd actually be healthier having constant low-level infection. So that argues in favor of the variances hypothesis. However, there's also some countervailing arguments. The studies he cited actually came up with 1 or 2 really spectacular hits, and he kind of dismissed those out of hand for reasons I found weak. There are other studies that he didn't give as much attention to that showed rather better hit rates. And there has been a review, a 2-part review published in 2013 and 2014 that reported on quite a number of compounds found in antimalarial African plants in African research that had very low inhibitory concentrations. Now, probably very few of those have been fully studied to see whether they might be made into drugs, because it costs many millions of dollars to prepare a drug for the international market. So I question whether the dearth of drugs Marantz points out is not, in fact, a dearth of funding. So the experience of the ICBG with regard to screening plants for malaria seemed relevant. They do a bioassay, which involves cultivating Plasmodium falciparum, chloroquine-resistant strain, in human red blood cells in culture. You incubate them for at least 1 life cycle, 48 hours, or you can go 2 life cycles, 96 hours, and then you stain with a cybergreen stain that interpolates into the DNA. Since mature human red blood cells do not have DNA, you then shine fluorescent light basically on this culture of red blood cells, and the more fluorescence you get, the more DNA is there, because the DNA all has to come from the parasite. You can calculate the quantity of parasite. And the results of screening assays by this method are reported as IC50s, or 50% inhibitory concentration. What concentration of your substance do you have to add to that culture to reduce the ultimate quantity of live parasites by 50%? Now, we had almost 1,300 samples of plants that have been randomly collected and run through this process. And I say random, this is one of the weaknesses of this. There's never such a thing as truly random plant sampling. but they were collected without any eye to their traditional uses. Once that had been done, post facto, we went through ethnobotanical literature and looked for reports of the use of those species or the genera to which they belonged for malaria. Now, some samples were identified only to the generic level, and some medicines in literature were only identified to the generic level. So we coded the use data both at the genus and the species level. And species could be coded as either definitely reported used, reported not reported used, or uncertain, where, yeah, the genus was used, but whether that species was used or not, we weren't sure. And here are the results. Now, in the way CNARP codes their data, the IC50 is, I'm not going to mess with that, but you can see up there the IC50 of less than 50 nanograms per milliliter, micrograms per milliliter is considered active. But those aren't really attractive enough to keep working on. They don't consider an extract highly active and worth further investigation unless the IC50 is less than 20 micrograms per milliliter. And the best ones are down around 2 or 3. Now, for a pharma drug, that wouldn't be impressive. Artemisinin is active in the nanomolar range. However, for a crude plant extract, it's very good indeed. You can imagine easily getting, if you think about how many liters of fluid there are in the human body, 2 micrograms per milliliter is something you could easily arrive at with a few cups of tea. So when we look at the whole data set, 39.6% of the samples were initially active and 21.1% highly active. Now, that has to do with the fact that you dump in a lot of something onto anything growing in culture and it'll kill it, right, eventually, but not all of these are really going to be attractive as drugs. They may have, for example, a very non-specific effect, or it may be just a lot of tannin in it, and so you have to try to sort out what's the relevant mechanism. But when we look at samples from general species used for malaria, you see modestly higher rates of activity. Samples from species that themselves had documented use, and this is a much smaller fraction of the data set, had a higher percentage of activity, but actually a lower percentage of high activity than the random data set. Now, there are limitations to this study. The plant parts that were screened included all parts of the plant, and not just the parts a traditional healer would have used. The extracts don't follow traditional methods, and we don't really know whether some of these samples, the particular species was used or not because of limitations in both the field and the library. So we're not saying anything whatsoever about the efficacy of these medicines in practice. However, our results do suggest that in this context, ethnobotanically directed screening would have been a good idea. It would not have increased the first-pass hit rates much at all, and it would have missed a large portion of the first-pass hits if we'd only used genera that were known to be medicinally used. We would have missed almost half the first-pass hits. If we had only confined it to those few species that we had identified in the field and knew that those species had been used, 95% of those first-pass hits would have been missed. So you lose a lot more than you gain by limiting the scope of your screening in that way, at least in this context. So I'm sort of disappointed to say that I haven't got any further evidence to use against Dr. Maren's hypothesis, which I don't like, but still can't disprove. And I'd like to acknowledge the federal government for funding and the government of Madagascar in particular for their support. Thank you.
We have a half a minute for one, two, three questions.
Thanks, Maria. It was a great talk.
My question is about limitations in the bioassays themselves that were mentioned, I guess, in this hypothesis. What do you think your results might look like if you were also looking at more of the host, so not necessarily the parasite, but also including a wider array of assays, if those even exist?
I think you would find a greater percentage of activity, but I don't know that the assays exist. They certainly aren't simple or cheap. This one assay, I thought it was interesting that the rate of sort of limited mild activity did go up. Maybe if you use something that's just semi-active, that's desirable to do this thing of knocking it down to a low level, because there are these places in Madagascar where you'll just have malaria all the time. But it's
Nice.